Sexo-reproduction in angiosperms. Life cycle of angiosperms involve two phases: diploid sporophytic phase and haploid gametophytic phase. The plant body, differentiated into root, stem and leaves, represent the sporophytic phase, which is the dominant phase in life cycle of angiosperms. At maturity, the sporophyte produces flowers. Haploid microspores and megaspores formed in the stamen and carpel of the flower respectively are the first cells of gametophytic phase. Microspores give rise to male gametes and megaspores form the female gametes. The diploid zygote, formed by the fusion of male and female gametes, initiates the sporophytic phase. Thus, gametophytic phase is extremely reduced in case of angiosperms. Sexo-reproduction. Sexo-reproduction in flowering plants, which are also known as angiosperms, involves the process of formation and fusion of haploid gametes, resulting in the production of a diploid zygote that grows to produce new individual. During this process, meiosis occurs, resulting in the formation of microspores or pollen grains in anther part of the stamens and megaspores in ovule part of the carpel. The phenomenon is called sporogenesis. Male gametes are transferred to the ovule by the process of pollination. After fertilization, ovule ripens to form seed and the ovary matures into fruit. Seeds after dispersal germinate to produce new plant. Thus the gamete formation, then pollination and fusion of gametes or fertilization, these are the main events in sexo-reproduction in case of angiosperms. This is the structure of flower. A typical flower, angiosperm flower, consists of sepals, petals, stamens and carpel. Do you know the functions of different parts of flower? Sepals are protective in function and they protect the flower in bud condition. Petals, they are brightly colored generally and they attract the insects for pollination. Stamens, they are male reproductive part of the flower, and carpel, that is the female reproductive part of the flower. Microsporogenesis. Stamens are the male reproductive part of the flower. They are collectively called androecium. Each stamen is consists of two parts, the lower sterile part filament and the upper fertile part anther. Microsporogenesis is the process involving series of events which lead to the progressive development of unicellular microspores or pollen grains. It takes place inside the microsporangia or pollen sac of the anther. Structure of anther. A typical angiosperm anther is bilobed structure. Each anther lobe has a pair of pollen sacs or microsporangia. Thus, we can say that each anther has 4 microsporangia. The whole anther is covered externally by a common epidermis. Microsporangium is cylindrical structure which appears circular in transverse section. It is consist of two parts, microsporangial wall and sporogenous tissue. This is the transverse section of mature anther, showing different wall layers as well as pollen sac. The wall layers include the outermost covering epidermis, which is single layer, then next to epidermis This is endothecium, and inner to endothecium there are a few layers which form middle layers, and the innermost layer of the anther wall, that is known as tapetum, which is nutritive in function. And the pollen sac, it contains the sporogenous tissue which later on forms the microspores or pollen grains. As far as microsporangial wall is concerned, the mature microsporangial wall is consists of four layers. The outermost layer, epidermis, is made up of tangentially stretched cells. Next to epidermis is endothecium, which is subepidermal layer of the anther wall and consists of radially elongated cells. Middle layers, there are 1 to 3 layers lying next to endothecium, and these are known as middle layers. Tapetum, the innermost wall layer is tapetum. It nourishes the developing pollen grains. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. The outer three layers, including epidermis, endothecium, and middle layers of anther wall, perform the function of protection and help in the dehiscence of anther to release the pollen. Sporogenous tissue. Microspores develop from the sporogenous tissue. It lies inside the microsporangium. All the cells are similar and are called sporogenous cells. These cells divide regularly to form diploid microspore mother cells. Each microspore mother cell undergo meiosis and form four haploid microspores which are arranged in tetrad. This phenomenon is known as microsporogenesis. As the anther matures and dehydrate, the microspores dissociate from each other and develop into pollen grain. Inside each microsporangium, several thousands of microspores or pollen grains are formed, which are released with the dehiscence of anther. Development of male gametophyte. Due to peripheral position of nucleus, the protoplast of the pollen grain divides unequally into two cells, the larger tube cell or vegetative cell and the smaller one, that is generative cell. Tube cell is also known as vegetative cell. Tube cell or vegetative cell is rich in vacuoles as well as reserve food. Slowly its nucleus enlarges and becomes convoluted. Generative cell is lenticular to spherical in outline. It has a thin layer of cytoplasm around a compact nucleus. A layer of callose develops around the generative cell. It helps in separation of generative cell from the wall of pollen grain. Later on, the callose covering of generative cell dissolves. In several plants, the pollen grains are shed in two-cell stage. But in many others, the generative cell divide further into two male gametes. In such cases, pollen grains are shed in three-cell stage. Megasporogenesis. Carpels are the female reproductive organs of a flower. They are collectively called gynoecium. Each carpel is consist of three parts: stigma, style, and ovary. Stigma is the upper broader region which is specialized for receiving pollen grains. Style is the long stalk-like structure. It is the median sterile part. Ovary, it is the basal swollen ovule containing region. Ovule or megasporangium. Ovule is attached to the placenta by means of a basal stalk called funicle. The region where the body of ovule fuses with the funicle is known as hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Ovule having one integument are regarded as unitegmic, and those having two integument are considered to be bitegmic. It encircles the ovule except at the tip, where a small opening called micropyle is organized. The basal part of the ovule opposite the micropyle, that is known as chalaza. The main body of the ovule consists of a rounded or oval mass of thin-walled parenchymatous cells called nucellus. Inside the nucellus, there is embryo sac or female gametophyte. This is the structure of ovule, having this basal stalk known as funicle or funiculus. Then this region region of the ovule which join the stalk or funicle with the body of the ovule, that is the hilum. This region is hilum. Then body of the ovule, that is having outermost covering, protective coverings called integuments. Then inner to integument there is mass of cells which is known as nucellus, which forms a nucellus, and inner to nucellus there is embryo sac. This opening end of the ovule, that is micropyle, and opposite to this end, this region is chalazal end. Megasporogenesis. Megasporogenesis is the process of formation of haploid megaspores from diploid megaspore mother cells. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. During ovule development, a subepidermal cell at the apex of the nucellus differentiates as archisporal initial. It either directly functions as megaspore mother cell, or it divides by a periclinal wall forming outer primary parietal cell and inner primary sporogenous cell. This primary sporogenous cell functions as megaspore mother cell. The megaspore mother cell undergo meiosis to form 4 haploid megaspores. Female gametophyte. In a majority of flowering plants, one of the megaspore is functional while the other 3 degenerate. Only the functional megaspore develops into the female gametophytes or embryo sac. The functional megaspore represent the first cell of female gametophyte. The nucleus of the functional megaspore undergoes 3 mitotic divisions to form 8 nuclei. The 2 daughter nuclei formed as a result of first mitotic division are separated by a large central vacuole so that the 2 nuclei move to the opposite poles. Both of these nuclei further undergo 2 mitotic divisions, and thus 4 nuclei are formed at each pole. Subsequently, one nucleus from each pole migrates toward the center. These 2 nuclei are called pollen nuclei, which later on form the secondary nucleus. The 3 nuclei left at the chalazal end are surrounded by walls forming antipodals. Among the 3 nuclei located at the micropylar end, one serves as egg and the other 2 as synergid. These 3, one egg and 2 synergids, together constitute the egg apparatus. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus. This filiform apparatus plays an important role in guiding the pollen tube into the synergids. The entire structure with 2 polar nuclei, 3 antipodals, 1 egg and 2 synergids constitute the female gametophyte or embryo sac. Pollination. Pollination is the process of transfer of pollen grains from anther to the stigma of flower. It is of two types, either self-pollination or cross-pollination. Self-pollination involves the transfer of pollen grains from anther to stigma of the same flower or to another flower on the same plant. When there is transfer of pollen grain from anther to stigma of the same flower, that phenomenon is known as autogamy. And if there is transfer of pollen grain from anther of one flower to stigma of another flower on the same plant, that process is known as geitonogamy. Cross-pollination is also known as xenogamy. It is the transfer of pollen grain from anther of flower on one plant to stigma of the flower on another plant. The various agencies involved in the cross-pollination are classified into two categories, either biotic agencies or abiotic agencies. Biotic agents include insects, birds, bats, snails, etc. And abiotic agents include wind and water. Pollen-pistil interaction. Once the pollen grains reach the receptive stigma after pollination, its germination starts. The stigma shows several adaptations to achieve this. It secretes sticky stigmatic fluid to which pollen get adhered. This stigmatic fluid contains lipids, sugars, resins, etc. and thus provide suitable medium for pollen germination. On stigmatic surface, pollens get hydrated, swell on absorbing water, they swell and develop pollen tube as the intine emerges out through the germ pore. As the pollen tube develops, the entire content of the two-celled pollen grain moves into the pollen tube. The generative cell divides to form two male gametes. This pollen tube penetrates into stigmatic papillae and then passes through the style and ultimately reaches the micropylar end of the ovule. Fertilization. Double fertilization was discovered in case of angiosperm by Nawaschin 1898 in Fritillaria and Lilium. After entering the embryo sac, the pollen tube releases its content in one of the synergids. One of the male gamete moves toward the egg cell and fuses with its nucleus. This process is known as syngamy, fusion of gametes. This results in the formation of diploid zygote. The other male gamete moves toward the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus. As this involves the fusion of three haploid nuclei, it is termed as triple fusion. Since two types of fusions, syngamy and triple fusion, take place in an embryo sac, the phenomena is termed as double fertilization. The primary endosperm nucleus develops into the endosperm, which is nutritive tissue, and provide nourishment to the developing embryo, while the zygote develop into embryo. The mature embryo is consist of two cotyledons in case of dicots and one cotyledon in case of monocots. It has an embryonal axis having plumule above the level of cotyledons and radicle below the cotyledons. When the seed germinates, the plumule give rise to shoot system and the radicle give rise to root system of the future plant. And with this, the life cycle of angiosperm that completes. Thank you.